What if life didn't have to end? What if cells could freeze, die, nearly die, but then they came back perfectly alive? Some organisms already do this, and the secret isn't magic, it's a sugar. What is it? Trehalose.
So what exactly is Trehalose, FARAH? I don't understand.
Oh, good thing you asked, MAISARAH. Actually, Trehalose is a disaccharide sugar made of two glucose molecules. Its chemical formula is C12H22 or 11, and it is mostly found in mushrooms, plants, insects, bacteria, yeast, and also used in food and medicine. So yeah, it isn't just another sugar, it's basically everywhere.
For chemical formula, Trehalose has a molecular formula of C12H22Co11 with a molar mass of about 342.30 grams per mole. It is classified as a disaccharide composed of two D-glucose molecules linked together. In terms of structure, Trehalose can be represented using several models. The facial projection shows the linear form of glucosinate, while the Howell projection illustrates the cyclic ring structure. The ball and stick model provides a three-dimensional view, clearly showing atomic bonding and spatial arrangement. Moving on to physical properties, Trehalose is a white, crystalline, odorless powder with mild sweetness. It is less sweet than sucrose. It is also highly soluble in water, non-hygroscopic, meaning it does not easily absorb moisture, and it has a high melting point of about 203 degrees Celsius, making it heat stable. For its chemical properties, Trehalose contains an alpha-alpha-1,1 glycosidic bond, which makes it a non-reducing sugar. So this bond provides high chemical stability, allowing the Trehalose to remain stable under the heat and acidic conditions. It is resistant to degradation and can be broken down into glucose by the enzyme Trehalase. So Trehalose has important biological roles. It functions as an energy storage molecule, supplying the glucose when needed. It also provides structural support by stabilizing proteins and cell membranes during stress conditions. Additionally, Trehalose plays a role in cell signaling, helping them regulate inflammation and stress responses.
The synthesis of Trehalose. The first one here reported in EU bacteria, archaea, and plants. It involves two enzymatic steps catalyzed by trehalose 6-phosphate synthase, which is TPS, and trehalose phosphatase, which is TPP. Next, the second one is reported in Parmulobacter sp, and these autologs have found in many other eubacteria. The third one is reported in Thermophilic archaea of the genus Sulfolobus. This organism synthesizes trehalose in two enzymatic steps, which are TRA and trans. And this is the fourth one. The fourth one reported in some fungi. There is uncertainty about the participation of the trapped enzyme in the synthesis or degradation of the trehalose. Why? Because the biosynthetic pathways is only shown in vitro. That's why. This one here is found in the hyperthermophilic archaeon Thermococcus litoralis. Mechanistically, the synthetic pathways works by forming a glycosidic bond between two glucose units, while stabilizing the molecules through phosphorylation first, then finalizing the glucose structure through dephosphorylation. And industrially, trehalose is produced from starch using controlled enzymatic bioconversion, making large-scale production possible for foods, pharmaceuticals, and biotechnology uses.
Trehalose is extracted from mushrooms and yeast used in beer and bread. It's only found in small amounts, accounting for only 0.5% of the total weight of a shiitake mushroom. Mass production was difficult and the market price was high, so it was used mainly in high-end moisturizing beauty products. However, in 1994, researchers at the company succeeded in extracting enzymes created by microorganisms in soil to make trehalose out of starch. This was the first time in the world that trehalose could be mass-produced affordably.
And lastly, this one here is trehalose biosynthesis pathway in fungi. And this one here shows a burnt trehalose used in food industries by Japanese company that consists of high purity of trehalose.
In terms of applications, trehalose is widely used across the industries. In the food industry, it stabilizes the food components and extends the shelf life. In pharmaceuticals and biotechnology, it acts as a cryoprotectant, preserving the vaccines, enzyme, cell, and biologics. In nanotechnology, trehalose stabilizes nanoparticles, improves drug delivery. In biofuels, it increases microorganism stress tolerance, enhancing the bioethanol production. It is also used in sustainable materials and green chemistry as a renewable, non-toxic alternative to synthetic fertilizer.
The health implications of trehalose.
Nutritionally, trehalose is an energy-providing carbohydrate supplying about 4 kilocalories per gram. Unlike other sugars, trehalose raises blood glucose more slowly, which may help reducing blood sugar spike. And you know what? Some scientists think it might help protect cells and even could be useful for studying brain diseases like Alzheimer and Parkinson's. Oh, but it is not that perfect. Eating it alone can upset your stomach and it can change the bacteria in your gut in ways that we are still trying to understand about it. So it can be helpful, but enjoying it in moderation is the best way to consume the trehalose, while research is still continues.
And finally, for recent research in 2024, reported enhanced trehalose production through enzyme engineering. A study in Francis microbiology showed that engineered enzyme from Bacillus species increased trehalose yield by about 32.6%, improving the industrial scalability and cost efficiency. Thank you.